Hello everyone, this is Dr. Nasib Medical Lecture. Today our lecture is about gametogenesis, meiosis and mitosis. So this is the chapter 2 of the general embryology. So gametogenesis means the conversion of germ cells into male and female gametes. Before we go through this process, we should know about the germ cell. What is germ cell? The germ cell is actually the 2n or diploid cell which contains the double copy of each chromosomes, one from father and one from mother. Gamete is actually the product of the germ cell which contain haploid number of chromosomes. So male gamete, which can also be called as a sperm, has 22 autosomes and one Y chromosomes, which is also called as a sex chromosome because it identifies the gender, determines the gender. So female gamete, which is also called oocyte, has the same 22 autosome but one X chromosomes. So the fertilization is actually the when the sperm and the oocyte combine, then they form zygote. This whole process is called fertilization. The fertilization takes place in the uterus of the in the fallopian tube of the female. The gametes are derived from the primordial germ cells, which are actually the epiblastic cell, that is the upper layer of the two germ layer of embryo at the second week of the development. So this whole process is how this whole process takes place. Suppose this is the embryo, this is the yolk sac, this is the two germ layers, epiblast and hypoblast. The epiblast, from the epiblast there are some cells which start migrating at the start of second week and come to the wall of the yolk sac. So at the yolk sac, from the yolk sac they migrate, and at the same time they also divide through mitotic division. It can be identified from these diploid number because they are dividing as well as there is the 2n number of chromosomes. So they migrate to the gonads at the 4th week. At the end of the 5 week they completely migrate to the gonads. When they migrate to the gonads, after that they start gametogenesis. Gametogenesis means that is the oocyte formation in the female and in the sperm formation in the male. So this process is taken place by the meiosis. Meiosis means this is the this such type of a division which result in the daughter cell contains single copy of each gene or haploid number. So this is actually, you can see the end of the 3rd week of a picture of an embryo. This is the amniotic sac, amniotic cavity or sac. This is the yolk sac, and this is the wall of the yolk sac contains the primordial germ cell in the yolk sac. So this is a clinical correlate with the primordial germ cells. When primordial germ cells do not migrate completely, so these are actually the pluripotent stem cells. They form the teratomas. Teratomas are actually the tumors of disputed pluripotent stem cell. They contain the variety of tissues. The chromosome theory of inheritance. What is the chromosome theory of inheritance? From this figure you can understand that this is actually the diploid number of cell uh, contain two copy each from one from mother and one from father, and these are the genes present on each arm of the chromosome. So this is actually the somatic cell. Yeah, you can also say it is a primordial germ cell. So there is linked genes. So the chromosome theory of inheritance says, suppose there are two genes which are present on the same arm of the chromosome, they tend to inherit together, is like this. This is the first point of the chromosome theory of inheritance. And the second, they say that each diploid chromosome uh, contain two copy, one from mother and one from father. So this is actually the chromosome theory of inheritance, and you can say there is a haploid number of cells. So the process of cell division are of two types. One is mitosis, one is meiosis. Mitosis takes place in all somatic cells, while meiosis takes place only in germ cells. Mitosis is the process where cell divide and give rise to two daughter cells that contain the genetically identical to the parent cell. Means they are genetically identical to the parent cell, as exactly as the parent cell was. So each chromosome cells receive complete complement of 46 chromosomes. Suppose if I cell divide, so if I cell divide through mitotic division, it will make exactly the same cell as I has before. If the epithelial cell of the skin divide, it will exactly has the same cell as it was before. So before a cell enter to the mitosis, it replicates its DNA. There actually the cell cycle. In every cell cycle there are three phases. One is G1 phase, one is S phase, one is G2 phase, and there is the M phase. So in G1 phase, the cell prepare for mitosis. In S phase, it replicates its DNA. So this DNA replication takes place in S phase of the cell cycle. So when the cell cycle enter into the mitosis, it start this process. This phase is called prophase. Prophase is actually what that the chromosome begin to coil, contract, and condense, and this is the beginning of the prophase. Each chromosome now consists of two parallel subunits, chromatids, which are joined through centromere, and then the metaphase. Metaphase is what that the chromosomes line up in its equatorial plate, and their double structure can be visible through a microscope. Each chromosome is attached by a microtubule, and here it forms the mitotic spindle. That is what mitotic spindle is, the centromere plus microtubule. It forms the and also centriole. These form mitotic spindle. Anaphase is the third phase. Third, that is actually each chromosome divide in this phase, and the migration of chromatids takes place to the opposite pole of the spindle. A telophase, the chromosome uncoil and lengthen again, and the nuclear envelope of each cell reforms, and there is the cytoplasm divides into two, that is cytokinesis. So here is the diagram. You can see that the first phase, which is called prophase. In prophase, you can see that there is coiling of the DNA chromosome, and this contract and they condense as well. In the second phase, that is prometaphase, the two parallel subunits are, you can see this red has two parallel subunits, this green has also two units are there, and these chromatids, and they join through centromere. That's the metaphase. They come to the equatorial plane, and they attach to the mitotic spindle, which are centrioles and the microtubule fibers.